Hi everyone, thank you for being with us today for our first webinar of the year and first webinar of our Changing Face of Cancer webinar series. My name is Sabrina Hanna and I'm the Executive Director at the Save Your Skin Foundation. In today's webinar, we'll be taking a closer look at clinical trials and what patients need to know about them. Clinical trials are an important piece in the treatment of cancer, but also of all other diseases and illnesses, and at the heart of all medical advances. But what are they exactly? What is their purpose? How do they work? How are they designed? Where and how to get more information about them? Who is eligible and why you should participate? This webinar is meant to educate patients about clinical trials, as well as answer any myths or fears about them. Dr. Natasha Kekri, Associate Scientist, Clinical Epidemiology Program at Ottawa Hospital Research Institute, will help us answer these questions. Dr. Kekri has been appointed to the Department of Medicine in the Division of Hematology within the Blood and Marrow Transplant Program at the Ottawa Hospital, effective October 2015. She's also an Associate Scientist within the Ottawa Hospital Research Institute and an Assistant Professor of Medicine at the University of Ottawa. She completed her bachelor's in science at the University of Windsor, then obtained her medical degree from the University of Ottawa. She then trained at the University of Ottawa in internal medicine and hematology, and went on to do a fellowship in stem cell transplantation at Dana-Farber Cancer Institute in Boston, Massachusetts, with a master's in public health from Harvard University. Her research is focused on developing early phase clinical trials and moving homegrown therapeutic strategies into patients. She collaborates with a number of local investigators and scientists in Ottawa, studying hematological malignancies and blood and marrow transplant recipients more specifically. Her research interests include improving transplant-related outcomes, specifically working to improve relapse rates after transplant. Her background in epidemiology through her master's has also created an interest in projects with an epidemiologic focus, including but not limited to decision modeling and meta-analyses. She also participates with a number of cooperative groups in North America, including the Center for International Blood and Marrow Transplant Research, the Canadian Blood and Marrow Transplant Group, the American Society of Hematology, and the American Society for Blood and Marrow Transplantation. Thank you for being with us today, Natasha. And just before I hand the floor over to you, I'd just like to mention that there will be a Q&A at the end of the presentation. And I invite you to send us your questions through the question box that's located to the right of your screen as they come to you, or if you would prefer to send them in at the end of the presentation. And with that, I give the floor to you, Natasha.
Great, thank you. Thanks for that introduction. So basically, I am a hematologist in Ottawa. I treat patients with blood cancers, specifically leukemia and lymphoma, but I have a very heavy research focus in both clinical trials as well as in the lab with preclinical work as well. So I hope that this overview sort of outlines what clinical trials are and how they can benefit people and why we do them and what goes into them exactly. So I have no disclosures or conflicts of interest to mention. So basically, I'm going to hopefully outline properly the steps that go into getting to a clinical trial to begin with. So research starts out with an idea somewhere by someone or by a group of people, and that goes through testing in preclinical situations with the lab and with animal models, followed by clinical trials. And clinical trial is just a very broad term that can mean all kinds of different ways of doing research, and I will sort of outline what the steps are just to get to the trial. Once we get to the trial, there are many phases that go into a clinical trial as well as different types of trials. I'll try to outline what those are. I'll outline how we design them and who runs them and who designs them, as well as the purpose of the clinical trials and why we participate in them and why patients and physicians should participate in them. And hopefully I will answer some myths and fears about trials, and in answering your questions, hopefully can give everyone a better sense of what all is involved from both the patients as well as from our point of view as the physicians. So I want to start by telling a very well-documented, very public story about a patient from the University of Pennsylvania. This is Emily Whitehead. She was a 5-year-old girl in 2010 who just a few weeks after her birthday came into the hospital feeling very sick and was diagnosed very quickly with a type of blood cancer called acute lymphoblastic leukemia. Emily very quickly had to start on chemotherapy because this is a very aggressive form of blood cancer, and she received therapy through the hospital for quite a few months. This therapy made her very sick, and she did have to come into the hospital over and over again. Unfortunately, about 1.5 years after starting her chemo, she had a relapse of her disease and had to get more chemotherapy. And usually when that happens, we recommend a bone marrow transplant, which is the most aggressive form of therapy that we have against leukemia. It's actually one of the most aggressive types of cancer therapies in general that we have in the world. And just before she was scheduled for her bone marrow transplant, she again had a relapse of her disease and unfortunately had to have the transplant canceled. She was no longer eligible to undergo transplant. So you can look on YouTube. There's lots and lots of videos of Emily's story. Her family says they're very honest about the fact that she was told that she really had no other therapies available to her and that chemotherapy hadn't worked and she had very little time to live. And at this point, Emily was only 6 years old. Her parents went looking for a clinical trial, knowing that Emily was out of standard options for her disease. And in contacting the doctors at University of Pennsylvania, she was able to enroll on a phase 1 clinical trial of something called CAR T cells, which are basically genetically engineered cells, Emily's own cells that have been genetically engineered to try and attack her leukemia cells. This had never been done at the University of Pennsylvania. It was very difficult to get approval because Emily is a child and it is difficult to get experimental therapies into pediatric patients. But she was offered the therapy and her family understood the risk, knowing that she really was out of other options. And in lots of contact with the physicians, she underwent the therapy. This therapy is very toxic and did make her very sick. She was unconscious in the intensive care unit for quite a while. She was very close to death while in the intensive care unit, and her family really did think they were saying goodbye to her. But not miraculously, but importantly, she, within a few days of that therapy, did start to turn the corner, and it was almost all of a sudden, you'll hear the physicians, if they talk about her, say that almost all of a sudden she basically turned around and responded. And Emily is sort of our poster child for this CAR T-cell therapy, which is still very experimental, but she is now 4 years cancer-free and is still doing very well. And she's one of the first people treated on this clinical trial. So I tell this story not so that you know anything about Emily or about leukemia or about CAR T-cells, but to say that new ideas do have to come forward, and hopefully patients will have new options in the future. But these families take on sometimes a big risk in trying these experimental therapies, but there are reasons why we do them and why we do clinical trials. And for Emily's sake, in the end, this ended up being very beneficial. And that trial has now expanded. That was a phase 1, and I'll tell you what that means in a minute. But that trial has now expanded into phase 2 trials and hopefully soon into phase 3, so that we can hopefully move this kind of a therapy, which is still experimental, sort of mainstream into the clinical practice. So how do we get there? How do we get from CAR T cells in the lab to actual patients like Emily? So first we have to do research in the lab. Someone has to have an idea and test it out in either cells or in all kinds of different assays and models in the lab, basically with cells that we collect from patients or cells that we collect from mice and other animal models, all kinds of different ways to see if there really is a signal that this new idea could offer some potential benefit to a given disease. And I'm not just talking about cancer here. This applies to all therapies across the board in medicine. Once we have some basic signal that maybe this therapy works, we often have to go to animal models. That's not always true, but generally is true, to prove that there's some evidence that this therapy can work in this disease in an animal model, and that it would be safe in those animal models. We then apply for application to either Health Canada or FDA or any regulatory board, depending on what country you're in, to justify and approve the use of this for the first time in humans. So that's a clinical trial application, a CTA, that gets put forward with Health Canada to determine if this is something that Health Canada thinks is safe enough to try in a clinical trial setting. The trial then, just like in Emily's case, goes to a first-in-human clinical trial, which is what we call a phase 1, and I'll explain that in more detail in a second. Then we move to phase 2 to 4 trials, and then eventually, if the new therapy seems to be beneficial without harming people, it will eventually get to approval by Health Canada for the use outside of clinical trial. And once that happens, generally speaking, the company or the people who were involved in generating that therapy will provide it as more standard of care, and doctors can then prescribe it to patients outside of the clinical trial setting. So those are the basic steps of how we get there. There is a lot that goes into each of these steps, and these steps can take months to many, many years to get there. So CAR T-cell therapy has been around, for example, since the 80s, but the original sort of constructs of those cells just didn't have any great signal to get them to clinical trial. More recent models about 10 years ago in the lab showed that maybe they do actually work, and getting to the first in human trial took almost a decade just to get it into patients. And now, even with the growing evidence that we have that maybe it works, we still have a lot of clinical trial and research to do before this would reach approval by Health Canada and FDA to really get this into patients as standard of care. So the process is quite long. I'll go through some of those steps in a little bit more detail. In terms of the lab and animal research, which we call preclinical data, this helps us determine the safety. It doesn't always tell us if there's a signal for efficacy, that it actually works, but it gives us a sense of whether we can really put this therapy into a human. And there are a lot of animal models that can mimic human diseases. Cancer is one of them. A lot of the cancer models that we have do have animal models that can mimic the disease to some extent. Mice are a very commonly used animal because their lifespan is feasible in terms of looking at outcomes. They also have responses to therapy and chemotherapy and drug therapy that, although it might seem odd, is somewhat similar to humans in terms of knowing how a mouse will respond, can sometimes give us some clue as to how a human would respond. And it helps us troubleshoot problems. So any unexpected findings in a mouse, we can then work up and find more details about before we then move on to a clinical trial in humans. This process for mouse models can take anywhere from, like I said, a month to a few years to many, many years, depending on how difficult the science is, the feasibility, the ability to show a benefit can take quite a bit of time. And last thing I'll say about preclinical work is that this is done in all kinds of centers, mostly in academic university type settings where people have access to labs. A lot of PhD and master students and postdoc people and scientists work on this, often in collaboration with clinicians and sometimes not. It can depend on the type of therapy. So that's mainly done in labs and academic centers. If there happens to be a signal in the mice and they can get through Health Canada approval to get to the first in human trial, then a phase 1 trial is designed. And a phase 1 trial is meant to determine safety. It's not meant to determine anything about whether this drug is really going to improve care for a patient. And although this can be difficult to understand sometimes, that we give patients these new therapies without knowing if it's going to help, it is done in a pretty safeguarded type of manner. So this bottom graph shows you that this is one of the classic designs of how we do these trials, but we would enroll 3 patients at a very low starting dose of the new therapy. If those 3 patients do not have any problems with tolerance of the therapy, meaning that it was safe, then we can escalate the dose to the next level. And usually the starting dose is not the therapeutic dose we're looking for. We start much lower than the dose that we would actually want to give patients in order to look for a safety signal. And we can keep advancing the dose until we reach a level where patients are having issues with tolerability, meaning that it might still be working, but if we were to go any higher, patients would no longer be able to tolerate. So we reach what's called a dose-limiting toxicity, meaning that we would stop at a level where we think that patients would not be able to tolerate the next dose of the treatment. And at that point, we might either enroll more patients or we might just stop the trial, or we might try to enroll more patients at the lower dose to see where the recommended dose for patients really should be. The tolerance can depending on what the study is and what the physicians and the scientists have deemed to be thought to be important tolerability issues. Sometimes it can be simple side effects that patients just cannot stay on the drug because of side effects. Sometimes it can be that the side effects were unexpected, so we would stop and readdress that. But there's all kinds of things that can happen in here, and this is where the unexpected is what we are waiting for. So if the lab research hasn't shown any side effects and we're seeing them in patients, then we halt this very quickly to try and figure out what might be happening in those patients. What could be the reason for the side effects? If we get through that and we find what we call our maximum tolerated dose, then we take that and we use that dose to design a phase 2 trial. A phase 2 trial now is still looking at safety with more patients at that tolerable dose, but we're also looking at effectiveness at that point, trying to determine if there is a signal that this new therapy actually might work for patients. At this point, it's still what we would call a one-arm trial, meaning there's no comparator group. That's important because it's hard to draw firm conclusions about a new therapy based on a phase 2 trial when you can't really compare directly to standard of care. And so sometimes we see data in the media and we see all kinds of reports all over the place actually about phase 2 trials, but they don't always give us a firm answer on whether the new therapy is really going to be beneficial to patients in the long term, because we don't directly compare this therapy to standard of care in this phase 2 trial. The phase 2 really is just trying to look for a signal that maybe this drug warrants further assessment or this new therapy. And the reason I don't use the word drug very often in a talk like this is because nowadays there are all kinds of cellular therapies for cancer and immunotherapies and all kinds of different ways of giving new experimental treatments, and they're not always just a drug off the shelf that we are testing. So the next thing we do is a phase 3 trial, and here is where we do bring in a comparator arm. And there's words that we attach to a phase 3 trial. Randomized is the most important part. People call them randomized clinical trials, RCTs, and that's because randomization randomly assigns patients to each group, meaning that it's not someone like me deciding, well, I think you would benefit from the therapy versus you who would benefit from standard of care. That would bias our results. So basically, patients get enrolled through a computer system that randomly assigns them to one arm or the other arm of the study. The other part that's included in most of these trials is blinding. And so single blind means the patient doesn't know if they're receiving the placebo, which is standard of care, or if they're receiving the new therapy. That would be single blinding. And double blinding, you can see in this cartoon, basically is the doctor also is not aware of what the patient is receiving. And double blinding is the most ideal because it does help to unbias the outcomes that are being assessed by the physician. If the physician knows you're getting a treatment, they might be more likely to, say that you're improving when you're really not. If a patient knows that they're getting the treatment, they are also more likely to report that they're improving than they're not. And that's been studied extensively in the literature and looking at placebo versus therapies and whether we blind or don't blind if it affects outcomes, and it definitely does. So it's important when feasible to do blinding. Now, you could ask, why don't we just blind every trial? But the reality is you can't do blinding for every single trial because it is not always feasible. Some therapies do require some extensive supportive care, and patients and doctors sometimes need to know if they're getting the therapy. Sometimes, especially in the cellular therapy world, it can be difficult to fake it because you get an infusion reaction from the new therapy versus you would have no reaction with the placebo, and that's hard to that can be hard to miss. So I think although we'd always like to do blinding and randomization, it's not always feasible in a trial, and when possible, though, it should be conducted if possible. So after a phase 3 trial, most of these new therapies, if there's a signal in the phase 3 trial and they clearly show benefit against the standard of care, these things enter into patient care. Hopefully Health Canada or FDA approves them. They can be given to patients outside of a clinical trial. But often we also conduct phase 4 trials, and phase 4 trials gives us the opportunity to continue to follow the long-term effects of some of these therapies. So even if we prove that these therapies work, sometimes we need to see that over the years we don't add any new risk to a patient. And although this is not needed to get to patients in clinical practice, it's still important so that we do not forget that these things are new therapies, and it can take many, many years for patients to show any long-term effects of a therapy. And so we do often conduct these trials to make sure that we don't forget that. It's sort of a quality control measure for these trials. It also allows pharmaceutical companies and the funding boards for drugs and other therapies to do some post-marketing analysis and determine the cost-benefit sort of these new therapies. And sometimes it's run by pharmaceutical companies, sometimes it's run by physicians. It really depends on what the outcome of interest is. Cost effectiveness is becoming a bit of a hot topic, especially with cancer therapies becoming so expensive. If we're going to introduce a new therapy that costs $500,000, for example, for a patient, you need to prove that you're really improving quality of life and years of life to make it worth the hospital system and the government system putting money into that therapy. So it's very easy for me to say that I think all leukemia, the new therapy, should be funded, but every physician could say that about every disease that they treat, and so it's very difficult when you have limited funds to know how to do that. We do have very strict regulations about how we bring new therapies forward and how they get funded. So how do we actually go about designing all of those different phases of trials? And there's lots of people involved. The clinicians, people like me and scientists, will provide the ideas or the concept for the most part. And we're also responsible for making sure patients understand when they're getting enrolled in a trial what the process is, and make sure that they provide informed consent. The statisticians are the people who sort of help us calculate the number of patients needed in the trial, how to run the trial, when to analyze the data, all of those sorts of things that make the data as robust as possible. The pharmaceutical companies and the government help us in funding these trials because they are not cheap. These parts take a lot of effort, a lot of coordination, a lot of clinic space, a lot of clinic beds, and it can cost quite a bit of money. So there's quite a bit of funding that is required for these types of clinical trials. And we always have coordinators who help with the regulatory and the ethics applications. They also help flag possible patients who could be enrolled in these trials. They are the go-to person for patients when they're on trial, so that patients always have access to someone that they can speak to directly if they need to come in or be seen or have any issue with the clinical protocol. And most importantly is probably patients. So we need patients to actually do the trials, but patients can also help in designing trials, and this is becoming much more common than it used to be. We are getting patients to provide input now on not only how we design the trial, but how patients would like to see the trials run. And the reason for this is that I'll explain a little bit later why we fail sometimes at getting the clinical trial, but some of the time it has to do with not having patient buy-in. Patients think that the therapy, for example, is too inconvenient, it's too risky, it's not a well-designed trial. Patients often have very useful input, and knowing those things at the time of trial design can be really helpful. So this is becoming something that we do more and more. I have a patient advocate on most of my clinical trials who we try to involve as much as possible prior to getting to clinical trial, just to help us with the design. And it's not always a patient who has the disease that I'm going to be using the trial for. It's often a patient who is a good patient advocate who can think about things that a doctor won't think about, like, you know, how many times they have to drive to the hospital, parking, funding for parking for the patient, how many pokes do they have to get, do they really understand the consent forms, all kinds of things that it doesn't necessarily have to be a patient who's sick and requires the treatment. It can often be a patient who's advocating for other patients. So a little more detail on how we design the trial. The statisticians will calculate things to give us numbers, and that is very important because it helps us determine the degree of confidence with which I can conclude something at the end of a trial. It also tells me how to safely do the trial. So if I design a trial and I randomize 10 patients to the new treatment and 10 patients to the standard treatment, and I think that, you know, 8 patients in one side do well and 9 patients in the other arm do well, I can't really decide then which treatment is better because I only treated 10 patients. I can't make any statistical conclusions about whether one treatment is really better than the other. So to go through all of this funding and activity and putting patients through trials, if we don't have the adequate number of patients per arm of the trial, we would not have any confidence in the conclusions that we reach. So it's very important to have people, including our statisticians, who can help us ensure that we get the number of patients needed to conclude safely and confidently with whatever conclusion we raise. Ethics is also really important. We have to go through very rigorous ethics of applications and approval in order to ensure that the harm done to patients is very minimal or none. And this is a very complex process. As you can imagine, in a therapy that's never been given in a person before, it's a lot of regulatory issues, rightfully so, to make sure that giving this to patients will be as safe as possible. And this can be done for all kinds of different things. So medications that are approved by Health Canada for one treatment but not for the treatment of another disease, we call that off-label use, and that often has to go through a specific ethics board to make sure that Health Canada has given us access to the drug for this new disease because they need to make sure that they believe that that's safe. We have to make sure that with ethics, patients who cannot provide consent, so sometimes children who can't read, or people who don't have the mental capacity to properly consent are advocated for, and make sure that if they are being involved in the trial, that they are not being sort of coerced into it, and that they really understand what they're doing. Therapies that have gone through rigorous preclinical testing will get to trial, but someone needs to check over that preclinical work and make sure it was done properly. And so ethics is probably the most useful part prior to a clinical trial, to ensure that everything we do is as ethical and safe as possible for patients and for everyone really involved in the trial. And I lastly want to say that physicians must obtain informed consent, which I think is hugely important. I think if you looked 50 years ago at clinical trials, patients were sort of willing to do anything the physician said, and I think that that is old school and is not going to apply to today's medicine, because patients should be well informed of what they are going through. And so to just tell a patient that this therapy could offer you hope is just not enough. We really have to make sure patients understand the potential risks and benefits of the clinical trial, what they're getting, and make sure that they can provide a proper informed consent prior to going through the new therapy. In terms of who runs the trial and where, the principal investigator is always a physician who basically is in charge of the clinical trial. So that means that it's almost always a physician who ultimately runs the trial. Even if the trial is being run over 100 different sites, there's one principal investigator who's ultimately in charge. But we usually will have a site investigator for each center that participates as well. And they have to make sure that the trial is always run through the ethical and regulatory standards that we meet at the beginning of the trial. There are lots of CRCs, or clinical research coordinators, who help us coordinate and find the appropriate patients for these clinical trials, and our nurses and the physicians have to help as well in identifying patients and consenting them appropriately to the protocols that they might be benefiting from. These trials, they can be done in both community and academic centers, and lots of cancer trials are done at both. But as patients will tell you, and as I've noticed in my own career, there are definitely more options available usually at an academic center. And the main reason for that is that the academic centers are affiliated with the universities, which are the places where some of these ideas are grown. So if the University of Ottawa has a scientist here who wants to bring something forward to clinic, I might do the first phase 1 and phase 2 trial within the Ottawa hospital and later on disseminate it to other hospitals. Bigger trials that are run through pharmaceutical companies can often be done across Canada and can be done at smaller community centers because it's a different protocol, it's a different type of therapy, a different type of trial, and in that setting, often community hospitals participate. I will say that because I treat most of Northern Ontario, I deal with a lot of the smaller community hospitals, and those patients also do have access to clinical trials. The difficulty there is that often these patients have to move or be able to get to an academic center where they can get these trials or these trial therapies, because often we have to train physicians and pharmacists and nurses for these new therapies in order to disseminate them, and so we can't always train people in every single center. We sort of pick sort of the bigger academic or bigger clinical hospitals to run those trials, and we refer patients into those centers. So I think that's basically my overview of what a clinical trial really is and how we get there. But the next part of this really is to say, why do we need clinical trials? And I think I've sort of made the case of how we do try to keep them safe, but we really need to think about why we are doing all of this. And the reason we don't just give these new therapies to patients one-on-one, which is not completely impractical. If you think about it, I could approach a patient and say, look, we have this brilliant idea in the lab called CAR T cells. I'd love to try it on you if you're willing, and make sure you completely understand the risks and benefits, and then take you through the treatment. That's not completely unreasonable if a patient and a physician both agreed. But the problem with that is twofold. One is that when we do one-off things like that, we don't learn anything for the patient or for the future. We can't really draw any conclusions from one patient. And secondly, it then is done outside of a regulatory body who can make sure that the study is safe and ethical. So it's easy for me, because I believe in CAR T cells, for example, to tell a patient, I need to give you this therapy. But I am obviously biased, especially if it's my own research. I would be highly biased in trying to convince a patient, you should do this. When it's done through a clinical trial, it gives us the opportunity to have external people looking in on this experimental therapy and telling us, is it safe and is it ethical and is it being done in an unbiased way? And I think that's probably the part that people sometimes forget about a clinical trial, is that it really does give us the opportunity to have outside people look in and tell you, is this appropriate for a patient? And the second part of that is because of that process, how difficult it is to go from lab research to phase 1 to phase 2, we over time lose the number of patients. And this graph is a very commonly shown graph for research, where we have these two death valleys, we call them, where basic biomedical research or lab research doesn't really get translated. As you can see here, the peaks decrease with time. It doesn't get translated into clinical knowledge or clinical use because over time, either the regulations around it make it too difficult for us to get there, but also physicians and patients, if they're not engaged in participating, we don't reach the ability to bring these newer therapies to clinical trial or to clinical practice. And so this curve just shows you that over time, we take a ton of ideas in the world and only about 10% of them actually reach clinical practice. And I think we could do better at that if we started engaging patients and physicians more in the clinical trial process and brought these ideas more often to the clinic. I think we'd have a better chance of finding new therapies and new cures for lots of diseases, especially cancer. So I think that sort of shows the barriers to getting us to trial. But why do we need them? I've already outlined the case for the ethical and the safety side of it, but there are other parts as well. I think I mentioned earlier about Health Canada that some therapies are approved in one disease and not another. This allows us to run, the reason to run a clinical trial here is to show that that therapy is still safe for a different population of patients. And it is sometimes difficult to understand that if Health Canada approves a drug for one group, why wouldn't they approve it for another? But patients, based on the types of diseases that they have, they really can be very different. So for example, in my blood cancers that I treat, a lot of my patients have no immune system from the therapies that I've had to give them, which would be very different than a patient who has a solid tumor who's only ever had surgery, for example. And so even though it might seem like two cancer patients should both have access to that therapy, they might be very different in terms of the type of patient they are. And so it's important that we test those things in the clinical trial setting where we can test safety and we can very appropriately watch over patients before we would just disseminate that to both patient populations. Another scenario where we might do a clinical trial, not just to show efficacy, is that some therapies may show a response, meaning that we know they work, and they might work just as well as another therapy. But the side effects of the two drugs might be different. And doing a clinical trial, comparing them head to head, can sometimes tell us which drug is better tolerated by patients, which drug do patients prefer, which drug gives us the same outcome but doesn't have the same harm to a patient. So those are sort of situations where we'd also want to do a clinical trial. So even where we call it, where there's no clinical benefit to one versus the other, they both seem to be similar, a clinical trial again can answer the question, which one should we be prescribing? And then lastly, as you already can assume, patients like Emily who are out of options and really have no further treatments available to them, they deserve to try new experimental therapies that might have a potential benefit. As I think all of you could agree, if a patient is out of options, it doesn't mean they should go on clinical trial, but they should at least have the opportunity to consider a clinical trial where they could enter into an experimental therapy that has the potential to benefit them. And for someone like Emily, in the end, that worked out quite well. That's not always true, and that's why we do all of these processes. But I think patients deserve the right to at least hear about clinical trials in that setting if they want to consider them. The reason, like I already said, we can't just do that without a trial is that we still have to make sure it's safe for the patient as much as possible. And I will say, for example, with Emily, after how sick she got and the things that were done to sort of make her better, protocols were changed after that to know that if patients got sick with that therapy, we had things in place to give these patients when they were having significant side effects from the treatment, for example. So we did help Emily, but we also learned a lot from her as a patient that could then change the safety of the trial for future patients. So there's other ways to do clinical trials. It's not just let's compare drug A to drug B or placebo to drug There's other types of trials that we ask patients to participate in that don't necessarily have a type of a medical intervention. Some of examples of those would be patient surveys, for example, collection of patient samples that we use in the lab, but we don't actually, do anything for the patient with. We just collect them to learn from. And patient data for clinical research, so looking through a patient's chart to learn what happened to them after they got a certain therapy. These trials don't necessarily need to go through Health Canada approval, and they don't require because they don't require a medical intervention, but we still do require patients to consent. If I'm going to do these things, a patient should be willing to do a survey. It's still voluntary. They'd have to be willing to get poked for us to take a patient sample for whether it's blood or tissue. And if I need permission from a patient to go through their chart for medical research. So we still need informed consent in these situations. But there's huge value even in trials where we don't have an intervention. So patient surveys have become very common because they help us determine the quality of the care or the quality of life a patient is experiencing from a new therapy. So even in some therapies that might improve the number of years a patient is alive, which is what most clinical trials look at to prove that the drug works or the therapy works, we want to know how that's affecting a patient. If that's making a patient bedbound at home and miserable, we need to know that, because it might then mean that it's not a therapy that is really improving someone's quality of life. And patients are the best people to tell us that. These surveys can also help us within our healthcare programs to improve care for patients. So getting a patient's perspective on how they're treated by their physicians, how they feel the drug is being administered, how the nurses are able to handle side effects and things like that, that sort of information from a patient's perspective can be very useful in how we deliver the healthcare that we're providing to patients, whether it's on trial or off trial. But once we have those surveys, we can use that information to really help improve quality care. For patient samples in the lab, there's lots and lots of reasons why taking samples from a patient, which doesn't really have much bearing on a patient for the most part, other than an extra poke, which can be, is not without its own risk, but for patients who are already getting blood work, for example, sometimes we ask for an extra tube of blood. Patients are usually quite willing to do that, and I find that that is also hugely beneficial. So a lot of the prognostic models that exist for cancer, especially in my world of leukemia and lymphoma, have come from taking samples of tissue and blood from patients and determining certain genetic factors over the years. As we've learned more and more about genetics, what factors determine a patient's risk of relapse, for example, or what factors determine what therapy they might respond to. So now with these genetic markers that we can look for, we can determine if there are new targets for cancer cells, for example, that we can treat with a certain therapy. That's true for rheumatoid diseases and autoimmune diseases as well, and all kinds of other areas where if we take samples from patients and create a big bank of patient samples and can then test out theories of whether a certain genetic marker has a new therapy available for it, or or if it tells us something about prognosis, that can be really useful information for patient care. And the last example I give there is something that we can sometimes take patient samples after they've received a therapy and find out what it's doing to a patient's immune system, what it's doing to their blood system. It can give us a much better understanding of the treatments that we're giving, which can help us in the future, both with side effects, but also with who to treat with these treatments. So I've already really touched on this, so I won't go into too much detail here, but patients and physicians don't always engage in clinical trials, which is why sometimes it can be difficult to draw conclusions about a new therapy and move it forward into clinical practice. I would say medical practitioners, especially outside of academic centers, are exposed to a ton of publications every year, and it can be difficult to keep up if it's not made publicly available. The new therapy is done in one academic center, but if I don't disseminate that information appropriately to caregivers and physicians who work in my field, then I'm not doing I'm not doing my field any justice in that new therapy. Sometimes you'll get trials that show different data. Some trials say that the new therapy is useful, some say that it's not useful, and then it can be very difficult to determine what the conclusion should be, and that can make it very difficult to bring something to clinical practice. And of course, our healthcare systems, especially in Canada, can't just look at clinical factors. They have to think about cost and regulatory issues as well, which can make it difficult sometimes to bring a new therapy forward. So just a few things to wrap up about myths and fears that I think patients and physicians actually think about clinical trials. So the first one I want to say is that patients often tell me, I won't benefit from the trial, only the next person will. But I really don't think that's true. Patients can be enrolled in trials for many, many different reasons, and they can benefit in many different ways. Firstly, often they get access to a new therapy that they would not have access to outside of a clinical trial because safety still needs to be tested. So even if we're unclear what the safety profile is for a drug, oftentimes we've got enough preclinical data that we are pretty confident that this is something we think could work for a patient, but we can't use it outside of a clinical trial till we learn more. So it does give patients the opportunity to have new options and new therapies that they would otherwise not be able to access. Patients do get the satisfaction of knowing they are advancing the medical field, and in patients who have diseases that are well known and have large patient populations and patient groups that advocate for them, patients do seem, at least in my world, tell me that they do get satisfaction out of knowing that they're advancing the field in their disease entity. And in clinical trial, patients get very different follow-up than outside of clinical trial. They are seen much more frequently, both by the nurses and the physicians. They get much more blood work, much more care. It can often be an inconvenience, but it also does mean that we are keeping a very, very meticulous look in on those patients, because on clinical trial, we have to watch for safety at all times. And that, it's just a reality of clinical trial that we're forced to watch a patient in a different way than we would watch a patient who's not on clinical trial. So they do benefit in that way too. If the drug is in clinical trial, it must be risky. This is a very common one I hear all the time. But the reality is, by the time a trial reaches the stage where lots of patients are being enrolled on the trial, the drug or the therapy has already gone through pretty rigorous testing. So outside of phase 1 and phase 2 trials, which are very small number trials, they only usually include 10 to 20 or 30 patients, the larger trials where more patients will get enrolled, by then we at least know the safety profile of the drug. And when we know the safety profile of the drug, it becomes sort of less risky and less unexpected, I would say. And so I think that it is true in some situations where a therapy can be risky and patients need to know that. But for a lot of therapeutic drugs and trials out there right now, patients, actually we know what the safety profile is. So it's not like we're going in blind in terms of what we expect to happen for a patient in terms of the side effects. I get asked, why can't I tell a patient, and also I get asked by physicians why we can't tell them if the patient is getting placebo, meaning just a fake pill, versus the new therapy. And as I've mentioned already, the reason for this is that we would lose integrity in the results we obtain if patients and physicians knew which therapy they were getting. And that's been well proven before, that when we blind a trial, the results are much more objective, and when it's unblinded, the results are just more subjective, and it leads to us drawing conclusions that we maybe can't draw. And that can be in both directions. On the one hand, we could draw the conclusion that a new drug is useful when it really isn't, or we might draw the conclusion that it's actually not helpful at all, but really it was. We didn't have the right design to prove that. And then, are there any safeguards in a clinical trial? So I haven't talked a ton about this, but in phase 1, phase 2 trials, I keep talking about the fact that we have to check safety, and that patients might see this as an undue risk to them. But the reality is, we do have a safeguard, this Drug and Safety Monitoring Board. So the DSMB is created for every new trial of an experimental therapy, where external reviewers who are not in any way involved in the trial do a review of every adverse event that happens. So that can be as simple as a fever from the chemo, or a fever from the new therapy, versus a patient who ends up in ICU with a bad outcome. This Drug and Safety Monitoring Board reviews every single adverse event as it comes in and determines if it's safe to continue the trial. So they look and see, do they think the adverse event was related to the new therapy? Do they think it was expected or unexpected? Do they think that it's enough of a reason to stop the trial? And their job is completely to make sure patients are given treatment as safely as possible. And the other thing is that even if you enter a clinical trial as a patient, at any point in time you should feel comfortable to leave a clinical trial. This should not in any way impact your care. And the same goes for refusing to do a trial if it is offered to you. That should not in any way impact the care from your physician. And there should, in no way should patients feel pressured to enter a trial by a clinician. And if they do feel pressured, they should try to find a way to talk to a different clinician or talk to the nurses and say that they feel like they're being pressured to enter the trial, because my job and our job as clinical scientists is to make sure that patients are entering trials in the most informed way and in a comfortable way where they feel like it is voluntary. Patients should not feel like they have to do these things. And like I said, if you leave a clinical trial, you still get the care that you're supposed to get from a clinician, even if it is outside of receiving the new therapy. So that's really all I wanted to say about what clinical trials are, and give you a chance to ask me questions. Just to, some last-minute summary slides here, summary points here is just that getting to clinical trials can take a long time. Once we reach trials, really the point of them is to make sure new therapies are safe and that we ethically bring them to clinic, making sure that patients are really the ones who will benefit from it, and not just that science will be advanced. And lastly, that patients, deserve access to new therapies, especially in situations where we don't have, cures yet, and that we pave the way for bringing these therapies into standard clinical practice in the future. So I'm sorry if I spoke too fast, but that's what I wanted to cover, and please feel free to ask me questions.
Thank you, Natasha. That was really great. I've already got a few questions, so I'm just going to get started right away.
Sure.
So when a patient is out of options, so the standard of care hasn't worked for them, other options haven't worked for them, especially in a cancer setting, when it comes to a metastatic or advanced stage, why is there a comparative arm in the clinical trials?
So that's a great question. So when patients are out of options, if the standard of care at that point there isn't one, so in situations like you said in cancer, when patients have relapsed or refractory disease and they've gone through all lines of therapy, we don't always do a phase 3 trial. So for example, even the CAR T-cell stuff that I was talking about and other immunotherapies that we're bringing forward for cancer patients who are really out of options, we don't do a phase 3 trial unless it's feasible. So if we can't do a comparator arm, we do what's called a historical control. So we enroll patients in a type of a phase 3 trial with enough patients that we get a robust signal about whether it's working. And then we compare to historical patients who have had similar treatments but didn't get the new therapy. So again, Health Canada would not approve that, saying that, giving patients, if you have a new option for a patient who now has no options, giving them no option versus giving them the new option, Health Canada would say that's probably not ethical. And so in those situations, we do often have to do one-arm trials where we then have to compare to the data that already exists. And unfortunately, that is the reality of cancer, especially when patients have been through 3 and 4 lines of therapy and they don't have any further therapy options. That is very common, and CAR T-cells is a classic example of that. The patients who we are currently giving CAR T-cells do not have any other chemotherapy options, and so it's going to be difficult to try and design phase 3 trials unless we have 2 different CAR T-cells or an immunotherapy that we're comparing to, or some other therapy that we can compare. We can't just offer patients nothing versus the new treatment. We try not to do that if it's unethical. Yeah.
I guess following along that, if you have a phase 2 clinical trial, so there's no comparative, so you know that it's the patients who are on it are responding, and so does that necessarily have to go, and I guess you sort of answered that already, it doesn't necessarily have to go into a phase 3 where there's a comparative?
No, it's just a numbers game. So phase 2 trials are meant to be small trials to look for a signal of efficacy, and often we'll do a phase 2 trial and realize that, oh, you know what, 80 to 90% of the patients responded. That looks amazing, but if you actually ask a statistician, they will say that you don't have any confidence to make that conclusion, even though, yes, 8 or 9 of the 10 patients responded. You don't have enough data there to say this for sure will work in a bigger cohort of patients. So even if you don't do a phase 3 trial where you're doing a comparator arm, you still need to do it in more patients to really prove with confidence that your conclusion really is the conclusion that you draw. And it's not just patients who have a hard time with that. A lot of clinicians have a hard time wanting to do that because, it's very easy to look at 10 patients, especially in the world I work in, where, when patients are out of options, we know what the outcome is. I know very clearly what the outcome is. If I see even 10 patients and see a signal there, I want to just go ahead and use it in everybody. But you need confidence in coming to a conclusion, because we have done this wrong in the past where we do phase 2 trials, we think we have a signal for something, and then we give it in a much bigger cohort of patients, and actually it really doesn't benefit them nearly as much as we thought it would. So there's good reason why we design these things with numbers with statisticians to really get a sense of, are we powered to say that this study has worked or that this new therapy has worked? And so that's the reason to expand it from a phase 2, even if it's to a bigger cohort of patients, not just to a phase 3 if you don't have a comparator, but really to give it to more patients before you go out and say we can give it to everybody.
Okay. I'm getting a lot of questions about eligibility criteria. So I was just at a conference, and the person who was speaking talked about the patients in the clinical trials as, it's sort of like the Olympic athletes in the Olympic hospital getting the Olympic treatment. And why is it that, the eligibility criteria is so,
yeah.
Yeah.
So it can be very frustrating for patients. And it's my day-to-day, I spend a lot of time speaking about that. And the reason is because things are in the media, patients have a very good sense of, they're well informed. And the reality is that if I'm doing a phase 1 or a phase 2 trial where I still don't know the full safety profile of my therapy, the only way to ethically try that new therapy is to give it to patients who have the best chance of not getting sick from the treatment. So for example, if I come forward with a new drug and I think this drug could cure cancer, for example, it's very easy to say, let's give this drug and see if it's safe. But if I'm giving that drug to patients to people who have never ever, a species that's never seen this drug before, I don't know what it's going to do to the liver. I don't know what it's going to do to the kidney. And to put patients in the trial who have kidney or liver disease or who are not completely healthy or who are not very fit, I'm doing them a disservice by starting out with my first trials including those patients. Not because I'm trying to prove that my drug is safe, but because ethically it's very hard to put a vulnerable patient into a trial with a vulnerable treatment, right? Where the treatment, we don't know what it's going to do to that patient. So it's not really the clinicians who design the trials who wouldn't want to enroll them. Trust me, I would love to enroll any patient on these treatments, but we have to do those first phase 1 and phase 2 trials have to be done in patients who are less likely to get sick from the treatment so that we can first determine, is it safe in them? And if it is, then we can expand it to other people. But to start with vulnerable patients and put them into those trials is really unfair to those patients because we really have no idea if this treatment will be safe in them, and they're already, at a bit of they're already vulnerable because they already have either, like I said, kidney and liver, sort of the common ones we talk about, or they're not fit enough, or their disease is actually at a stage where they could still benefit from standard of care, for example. I wouldn't want to put those people through an experimental treatment when it's in its early stages. And then other reasons come up as time goes by. So if in a phase 1 and phase 2 trial we find that this drug actually hurts the liver, then I'm never going to want to give this to patients where I think the harm outweighs the benefit. So it's very easy to say this drug cures cancer, but if this drug makes your liver fail and I can never get you out of that, I haven't done you a service by giving you that new therapy. So sometimes we find that some of these criteria we do have to keep sort of consistently throughout the trials. And that is, again, because we've determined safety-wise, we're losing the benefit when the risk is too high. And so our job really is to balance those two factors.
So I've got more questions related to eligibility criteria. So we have, in melanoma, specifically in the adjuvant setting, there aren't really any options. So it's, you're sort of waiting or,
yep.
But you're getting treatment so that it doesn't progress, but then that eliminates you from getting into clinical trials.
Mm-hmm.
And then we've also got another question here who's got a blood genetic disorder that also impairs him from getting into clinical trials. And he's wondering, how does he get access to these new therapies if he can't get access to the clinical trials?
Yeah, unfortunately, that's why we do clinical trials, is because we don't want to include people who could get it once we have really robust data. So again, when we get through phase 1, phase 2, phase 3 trials, we have plenty of patients enrolled. Health Canada can say, you know what, this therapy looks very safe and it definitely has a benefit to patients. Now you can give it to whoever you'd like. But again, until I have that data to prove that a therapy works, I can't just let everyone get access to it because I don't know the risk in those people. So someone who has a genetic condition, and this happens to us also in the leukemia lymphoma world, if they have a genetic underlying reason, we don't know in that genetic situation what this new therapy might do. And that's the problem. And a lot of the new, even in melanoma, a lot of the new therapies do manipulate the immune system, for example. And so you can't give this therapy necessarily to people who, for example, have an impaired immune system or have an active infection, or who might have underlying conditions where we have no idea what the crosstalk will be between this drug and those conditions, not in the early stages. So unfortunately, patients not all patients will have access to these therapies in the clinical trial setting. But the reason we do these clinical trials is to eventually disseminate these therapies to everyone who we think could benefit from it. And I agree, patients who have underlying conditions eventually, will eventually get a trial of seeing if these therapies will work, but that just is hard to do up front, and that's just the practical side of clinical trials, unfortunately. And the first question you asked me was, if I've been on one therapy, I'm no longer eligible for the next. It's very tricky in cancer, and even in melanoma, because if you've been on certain therapies, you lose eligibility for other therapies. But the problem is, it's easy to think that one therapy is better than the other, but we don't know that. So just because a new clinical trial becomes available and a treatment now becomes available doesn't mean that treatment is the right thing for every melanoma patient, for example. And some melanoma patients, yes, we want to offer you that drug, but if there's other therapies out there that could keep your disease at bay or could treat your disease, and they're known therapies within the world of melanoma, we would still want to offer those to you. So clinical trials are great, but we have to remember we do have standard of care, and in those standard of care, we know what those treatments are. They're very predictable. So a bone marrow transplant, for example, is a very risky procedure, but it is very predictable for me what to tell a patient about the risk and benefit of a bone marrow transplant. In CAR T cells or other new therapies in leukemia, I don't know what the risk benefit is yet. So I still think patients, should be offered a transplant where I know where they know what they're getting into. And yes, in some trials, if they've had the transplant, that no longer makes them eligible for CAR T cells in the future. But patients still deserve to be getting standard of care. And in fact, any doctor who takes them out of standard of care just to put them on clinical trial is doing a disservice to a patient, because that clinical trial still needs to be done before we know, what the signal or what the efficacy is going to be. So it's very frustrating for patients. I completely understand when they need to get on other therapies, but then that makes them not eligible for newer therapies. But again, it's because standard of care still should apply. Like, things that we know, what we know about, are still therapies that, we try to do things that are evidence-based, and those are things we have evidence for, if that makes sense.
Yeah, well, I think in melanoma it's a little different because standard of care previously was chemotherapy, and we knew scientifically that chemotherapy didn't respond to melanoma. So it was important to get melanoma patients into a clinical trial. But I think now with the new immunotherapies, that the standard of care is far more advanced than what it used to be.
Yeah, and those therapies that melanoma patients are now getting access to, don't forget, are also in other solid tumors like lung cancers and things like that. And it's the same debate there. We've known for a long time that chemo and surgery doesn't keep these patients, depending on the type of cancer, necessarily at bay, and we definitely knew that other options had to come. But it's easy to say that. It's another thing to actually go through regulation and make sure that the new therapy is not harmful, because it's now easy to say in hindsight for melanoma that these checkpoint inhibitors, for example, and the new immunotherapies have done wonders in this disease. But I can tell you, when checkpoint inhibitors started, we had no idea what would happen to the immune system when we gave those things. So it's easy now to look back and say, well, we should have been offering it to everybody, because we should have, but we didn't have the clinical trials to sort of show us that, and so that's why melanoma therapy has drastically changed over the last 10, 20 years, I think. Or maybe not that long, but I think that's part of it. Yeah.
So I've got just a couple more questions. When does unblinding occur in the phase 3 double-blinded trial? And also, how do patients get placed into the whatever group they're getting placed into? Is it just luck of the draw, or is it,
Yep. So how does randomization happen? How does unblinding happen? So randomization happens by a computer program, and we tell the computer what to randomize on. So if I'm enrolling patients, most studies we will randomize based on age and gender, and maybe one other thing. So the computer looks at your age and gender, and then based on how many patients have already been enrolled, decides which group you should end up in. And that's so that in the two arms we end up balancing the patients, so that instead of it being, oh, maybe the drug works, but the group who got the drug is actually all 20 years younger than the patients who didn't get the drug, it allows us to balance the two groups so we don't have to worry about any confounders conflicting my conflicting the conclusions that we raise. So that's why we do it, and that's how we do it. But it is a computer system that decides that. Sometimes if a disease has a lot of subcategories, so in leukemia, for example, we would have, or not leukemia, even in any disease, I guess, let's say leukemia then, if I had 2 different types of leukemia that I'm treating with this new drug, ideally I want to balance the 2 groups to have about the same number of patients of each type of leukemia in the 2 groups. So sometimes we'll randomize on age, gender, and the type of leukemia so that we can sort of balance the groups. Same with melanomas. If you're enrolling patients as frontline therapy and as relapse therapy, which would be exceedingly rare, but let's assume that that's happening, you want to balance the group so that not all the relapses are in one group versus the other, because that will change the conclusions you can really draw from the study. So we tell the computer what factors to randomize on, and then the computer spits out which arm you get entered in based on how many patients have come in the trial before you. That's randomization. In terms of unblinding, that's an excellent question. It really completely depends on how the physicians design the trial. Almost always you get, if you want to, you get unblinded after the trial has ended. So once the final patient has gone through the last follow-up of the trial, then we will unblind patients and physicians so that they both know what they receive. But it's usually at least 6 to 12 months after the trial, after the last patient is enrolled, because often, especially in cancer trials, we keep these patients on trial either for the rest of their life or for at least, you know, a couple of years. The outcomes are often like 1-year survival, 2-year survival, 5-year survival. So it can be a long time before we get unblinded. Having said that, if there's a safety concern and there's a reason to know, a physician can contact the principal investigator of the trial to unblind if there's a reason to know. For example, if a patient had an anaphylactic episode and needs to find out if they're allergic to the drug or allergic to some component of the drug or something like that, there are ways to unblind if we think it is for patient safety. We will unblind for those reasons, for example.
Okay, well, it is 3:01 now, actually, and maybe you can just a last word on getting more patients engaged in clinical trials.
Yeah, I think, you know, everyone who's on this call obviously, you know, thinks about this and is interested in it, and I would tell you that keeping engaged in the process of clinical trials and being involved in them is really important. I think, you know, my job as a clinician scientist is to advocate to both patients and physicians to really think about what clinical trials are available and make sure that patients are aware of them. I think it is still, unfortunately, very much a regulatory issue, and therefore I do understand all the frustrations about getting access to a new therapy, getting onto clinical trials, and all the issues with eligibility criteria. But remember, a lot of that is set up to make sure these therapies are safe enough to keep giving. And I'll conclude by saying that, you know, the way of the future and the way we're going to cure most of these diseases in the future, and hopefully in our lifetime, in my lifetime, is that I think we will need to, you know, make sure these clinical trials do continue to happen and that patients continue to be enrolled, and once clinical trials are done, that we then bring these things to clinical practice for everybody to receive access to them. And that's really going to be the only way that we change the face of some of these diseases, including things like melanoma and cancer.
That is so true. Well, thank you so much, Natasha. And for everybody who is on the line, the recording will be available on the Save Your Skin website, and you will also be able to come back to the Save Your Skin website and find out about our next webinar, which will be in March. So thank you to everyone who was on the line. Thank you so much, Natasha, and we'll be seeing you shortly.
No problem. Thank you.
Bye.